Enterotube test is an example of a rapid multitest system used in identification of unknown oxidase-negative, gram-negative rod-shaped bacteria of the family Enterobacteriaceae. Here we have the enterotube, which has 12 compartments and a pure culture of bacterium to use as inoculum for the test. When we look at the tube, there are two ends. In one end is a blue cap, in which there is a little handle to pull the wire that goes down the center of the tube. And as we can see, the wire goes through the center of the tube. On the other end is a white cap that covers a sterile needle, which is used to pick up a small amount of the bacterium to be identified. Each compartment has different colors and different biochemical tests. Here we can see that there are waxes on top of them. These waxes allow for the creation of an aerobic environment. The procedure begins by taking the Petri plate with a pure culture of bacterium. Then we are going to gently rub the needle on the colony to gather some of the bacterial sample on the end of the needle. Now we're going to go to the other end where the handle is and pull the wire all the way to the last compartment, applying a turning motion. Then, we place the wire back into the tube. Here we can see a little indentation in the wire. We break off the wire at that indentation. Now we can screw the caps back on the two ends. Next, we can see these little openings on the side here that are covered with plastic film. By using the part of the wire, we can carefully punch holes on the film. These little holes will allow oxygen in to aerate the compartment. Once that's done, we can label the tube for incubation. During incubation, the flat surface of the tube must be facing down, so the tube doesn't roll and no condensation will form on the inside of the plastic film. Now we can incubate the tube at 35 to 37 degrees Celsius for 18 to 24 hours. Over time, we will begin to see changes in color. Here we can see that the upper tube has not been inoculated, and the different biochemical tests are also labeled. Let's start by looking at the left-hand side. The test is glucose fermentation and the color change is from red to yellow. Again, the test is covered by a wax layer to provide for conditions that will trap any gas that is evolved. This indicates that there was no fermentation in this compartment. The yellow color change indicates that acid had been produced. Meanwhile, lifting of the wax indicates the production of gas. This can be proven by the appearance of a pocket or a mirrored surface. Next, there are 6 different carbohydrates that will provide a positive result with a color change to yellow from original reddish-orange. A modification of the color to a bright red is caused by deamination and is a negative result, whereas the second and third chambers will turn a deep purple for a positive result, and the fourth chamber has a medium that will indicate sulfur reduction by the presence of a black color. The ninth chamber provides for a double test of dositol, which gives a positive test as a sugar, or phenylalanine, which gives a positive test of a dark brown color. This chamber can be positive for either test, but not for both. For the urease test, a bright pink color will indicate a positive result, and the last chamber will turn brilliant blue for a positive result for citrate test. The remaining tests are visualized by the addition of reagents. Adding Kovac solution to chamber 4 or Barrett's A and B to chamber 9 can react with a chemical test that is produced by the bacterium. A negative reaction is shown by no color change, while a positive reaction is shown by a change to a red color by the reagent. After observing the changes, we can now read the result of the tubes. For example, the first chamber is positive for glucose, and inspecting the wax layer, a lifting of the layer is visible, hence it is positive for gas. Next, there is a black coloration for sulfur reduction, a dark brown color for phenylalanine, and a pink coloration for urea reduction. Adding the Kovac solution caused a cherry red color to form, which is a positive result for indole production. Now we can add up the numbers of the positive results and come up with a 5-digit code. This 5-digit code is then used to look up in the interpretation guide or code book the identification of the bacterium we tested. So from there, we have shown how to rapidly identify the specimen by a multitest system. Thank you so much for watching!